Yes, everyone, very warm welcome to the session. Very good evening, all. So this is the day 3, and today we are going to learn about the MCQs in immunology. So this will be of great help for your preparations into NII. And this is Dr. Saranya. I am talking to you on a very important session for your upcoming NII PhD entrance examination. Yes, everyone, I hope that you are all ready. Okay, so you can follow me at Unacademy and you can learn a lot. And definitely, if you are interested to come up with a subscription, please do use my code Saranya Live. This is little bit about my previous research work. Yes, so I was working with a biomaterial, as I told you earlier. Yes, so these are my research papers just for your inspiration. Okay, yes, everyone, good evening all. Yes, Sati, yes, Shishmita. Okay, good. Okay, so this is the batch course that is now on. Okay, so now I am taking developmental biology in the batch, and I am doing methods in the individual course. All right, everyone. Okay, so come on, everybody. Let us pay attention to the today's questions. Shall we do it? Yes, you're all ready for it. Okay, let us begin with a very simple question. Okay, right. Yeah. So, which one of the following is a primary lymphoid organ? Yeah, so the lymphoid organ, there are two types. Yeah, so one, it is going to be the primary lymphoid organ, and other one, it is going to be the secondary lymphoid organ. Correct? Yes, everybody. So shall we try to find out which of the following is a primary, and which of the following is a primary lymphoid organ? Yes, all. Good, everyone. Come on, keep it up. Yes. So is it going to be your lymph node, spleen, Peyer's patch, thymus? So which is coming under this primary lymphoid organ? Yes, all. So primary lymphoid organ, it is none other than the thymus, isn't it? So thymus is, just a minute, guys. Am I there? Am I connected? I couldn't see your responses. Just a moment. I'll just check whether I am there. All right. Yes, everyone. Good. Yes, all. Now I can see you. Yes, Shishmita, Sati, Sahil. Yes, Sati. Yes, Rishabh. And Madhu. Good, everyone. Correct. Yes, Shrishti. Yes, so this is none other than the thymus. Okay. Right. Yeah. So thymus, it is going to be the primary lymphoid organ. So what are these? What about lymph node, Peyer's patch? So they are all, what kind of organs they are? So do they come under primary? No, isn't it? Yes. So these T cells, they are actually derived from, Sushmita, they are secondary. The T cells that are derived from the bone marrow stem cells, and they will only get immunocompetent only in the thymus. So thymus is becoming a primary lymphoid organ, and all others, they are going to become the secondary lymphoid organs. Understood? Okay, good, everyone. Keep it up. Right. So these tangible or tingible bodies that are present inside the macrophages. What are they? What are these tingible bodies that are present inside the macrophages? So are they the DNA fragments? Or are they the phagocytized foreign antigen, or they are the sign of apoptosis if tingible bodies are present, or do they represent this bacterial wall component? What are these tingible bodies that are present inside the germinal center of macrophage? Macrophage is nucleated or not? Think. Macrophage, do they contain DNA? See here, I'll tell you, this is a very important point. Yes, it is none other than the DNA fragments. Okay, yes. So what happens is that, so if at all there is going to be this tingible body, it represents this cell death. Okay, yes, Sanjay. So it is going to represent this apoptosis. Okay. So when the apoptosis is taking place, what will happen to your intact DNA? The intact DNA, it is going to chopped. Okay. So it is going to get chopped and it is going to form DNA fragments. Okay. Yes, all. So what happens? There is going to be an extensive apoptotic death. Yes, so because of that death, it is going to be become fragmented. So this fragmented one, it is what it is called as the tingible bodies. Okay, yes everyone. So now you understood it. Yes. Okay, so when will these tingible bodies be present? The tingible bodies, they are present only during the apoptosis. Okay, so that is going to lead to the fragments. Alright everyone, this is clear? Yes? Okay. So this is also a very basic question. Which of the following lymphoid tissue is unencapsulated? They do not have an encapsulation. This is simple, no? Yeah. So which of the following it is going to be unencapsulated? Yes, all. Come on. So which of the following is going to be unencapsulated? It is going to be thymus, it is going to be lymph node, it is going to be spleen or malt. Yes, Sakshi. Good. No, I mean answering is good, but yeah, good. Yes, everybody, come on, you can give your answer. Which is going to be unencapsulated? That means it doesn't have any capsule. Yeah, yes, I'll tell you, this is going to be malt. Okay, yeah. So what happens is that, yes, yeah, Shushmita, yes, Sati, absolutely correct. Yes, so the correct answer is going to be this malt. Option D is the correct answer. Alright, yeah. What is this malt? Malt is nothing but it is going to be the mucosa associated lymphoid tissue. Okay, so what is this malt? This malt is going to be the mucosa associated lymphoid tissues. Right, yes. So what they do? They will make a small diffusion. Okay, but they are the non-encapsulated ones. Okay, yeah, they are called as diffuse. Yes, Mukhtar, very happy to know. So it is going to be non-encapsulated. Okay, which one? This malt. Okay, so the malt, they do not contain any kind of capsules. They are unencapsulated or they are non-encapsulated. Right? Yes. So they are, yes, Nenka. So they are, so they do not contain anything. So what is included under the malt? The malt is going to include this gut alt, that is the Peyer's patch, okay, as well as this bronchus related one, right? Yes, and this urinary system. Together they occur as your diffused lymphoid tissue. Okay, right? Yeah, so this malt, it is absolutely going to be non-encapsulated. They don't contain any kind of capsules. Yes, everyone. So now it is clear. Yeah, so we will make a move towards the next question. Okay, so we are practicing the MCQs today for the upcoming NII examination, and this will be helpful for your NET also, because in NET also we do have this immunology. Okay, so coming to the next question, and GATE also, BET also, any exam it will be helpful ultimately. Correct? Okay, so the binding of antigen to this antibody. If at all an antigen is binding with an antibody, what is it cells? Yeah, Shrishti, absolutely. So I told you, no, so everything, this Reut, it is a very important book. Yes, apart from QBA, the Reut MCQs, it will be very helpful for your especially this NII entrance examination. You will find this to be absolutely a ditto questions, like how we get Gilbert developmental biology for your CSIR net, right? Likewise, this Reut questions will be there. Okay, right? Yes, everyone, come on. So this binding of antigen to the antibody, what it will do? It is unaffected. Yes, Srishti. So it is unaffected by molecular rigidity. It is unaffected by the presence or absence of water molecules. It involves the covalent bonding. It is optimized by the spatial complementarity. Very good, everybody. Absolutely correct. So it is going to be the spatial complementarity. Correct. If it is going to be covalent, so then how will be the interaction taking place? It is very difficult. No. So they do not undergo the covalent. They will undergo only the non-specific interaction. Okay, yeah. So what are the non-specific interaction? Like van der Waals, yes, so all those things. Okay, so the spatial complementary, it is involved for the binding. Otherwise, this non-specific forces, they will depend on the reciprocal, isn't it? So it will depend upon the distance. So then only they will become strong. Okay, right? So absolutely it is going to be, yes everyone, yes Sati, yes Shrishti, yes Shushmita. So now you got it, everyone, right? So it is optimized only by the spatial complementarity. It is not because of covalent but due to the spatial one. Okay, so yes, shall we gradually make up a move? Okay, good. Yes, Shushmita, that is okay. Right, yes, that is okay, correct. Okay, so before that, shall we make a recap? Yeah, so what questions we have seen so far? Shall we just make a recap? Yeah, few questions were quite new and few were very familiar. Okay, just we'll make a recap on our memory. Okay, so which one of the following is a primary lymphoid organ? Yes, so primary lymphoid organ, it is going to be, this is how you have to revise the MCQs, okay? Right, so like how you were revising with like how you were revising with your theory, with your MCQs also you have to make up a revision. Yes, all correct. So primary lymphoid organ, it is going to be none other than thymus. So these Peyer's patch, these and all are coming under the secondary one. Okay, right, good everyone, keep it up. Right, so the next question, what about this tingible bodies? Okay, the tingible bodies that are present inside the germinal center. Yes, that's okay. Inside the germinal center of the macrophages are. So what did we saw about the tingible bodies? The tingible bodies are none other than the DNA fragments. So when will be these tingible bodies formed? They will be formed whenever there is going to be the apoptosis, right? Very good. So during the apoptosis only they will be formed. So that is about the tingible bodies, right? Fine. And the next question, yes, which is going to be the unencapsulated lymphoid tissues. Okay, so all are lymphoid tissues, and no, they have agreed with that, but they have asked which of the following it is an unencapsulated lymphoid tissue. Okay, so it is going to be none other than the malt. Very good. So malt is only going to be this unencapsulated lymphoid tissue. So this is going to include the mucosa associated malt, that is going to be malt. And then if it is going to be Peyer's patch, it is digestive system associated, and if it is going to be respiratory associated, it is the bronchus ALT. Clear everybody? Okay, and then we saw the question, the binding of antigen to the antibody. So this is going to get optimized by the spatial complementarity reaction. Done, right? So till this it is clear. Okay, so which of the following statements is incorrect? Okay, so the following statement is not at all correct. So which of the following is going to be the incorrect one? Right. So the first, it says that, what it says about the affinity. All right, fine. So it says that, yes, what is this affinity? The affinity is the measure of strength of binding of an antigen to this antibody. Right. Yeah, very good. And the next one says that it is the association constant of this antigen antibody equilibrium. Okay, right, good. And third one, it talks about this avidity. Good. And fourth one, it is the related to this free energy. Very good everyone, that's great. True. Yes, Sanjay, Anjali, Sati, Shushmita. Yes, Firades, Tanya. It is going to be none other C. Okay, so what is this avidity? Affinity and avidity, they are different, isn't it? Yeah, affinity means it is only one. Okay, avidity means it is the collective. Okay, so avidity, it is going to be the collective affinity, right? So if all the affinity it is collected together, it is going to form this one, right? Okay, so this affinity, it is related to the free energy change of the interaction. This is also correct statement, no? Obviously this antigen and antibody, if they are going to interact, it is the reaction. Yes, so if it is going to be the reaction, yes, so it is related to the free energy change. So this is going to be a spontaneous reaction, isn't it? So if this antigen and antibody, they are complement to each other, so they will bind and they will form an interaction. So that is going to be quite spontaneous. Got it, everyone? So they are asking which one is incorrect. So which one is incorrect? Affinity, it cannot be the avidity. It is the summing up effect is called as the avidity. Yes, everybody? Right? So shall we still move into an in-depth? Okay, right. So the processing of the cytosolic protein, okay, it is involving which of the following? Yes, Anjali. So the processing of cytosolic protein. Okay, right. So it is going to involve the transport into the late endosomes, proteasome mediated cleavage, displacement of the invariant chain, displacement of beta 2 microglobulin. So what will actually happen? Yes, Shashmita? I'll tell you. Okay, don't worry. So it is going to be a, you have heard about tar? Yes? No. This is going to be a proteasome mediated cleavage. Okay? Yeah, I'll tell you how it is taking place. So this is going to be the cystole or cytosol. Okay? Right. So this cytosolic protein, actually it should have been degraded by the proteasomes. Then only it will form the small ones. Now here who is present? This is the tar. What is this tar? What is this tar? Transport, isn't it? So these are the transporter associated protein. Okay, TAP, transporter associated protein, TAP are there. Okay, so this TAP, it cannot bind the big ones. So this TAP, it can bind only the broken peptides. So how will be the peptide broken? It will be broken only by the proteasomes. Okay. So absolutely you require the proteasomes. The proteasomes is breaking the peptides for you, and this peptide will bind with this TAP. It is the transport associated protein. Yes. So yes, Menka, I will zoom it. Which one you want to zoom? I will zoom it for you. Don't worry. This one. So this is an example. Okay. So this is present in the cytosol. Yes. And this is the TAP. Okay, everyone. Right. So now you got clarity. So how this cytosolic protein, they are getting processed, and how it is going to make a move inside. Okay. So it is going to undergo the proteasome mediated cleavage. Yes, everyone. Okay, that's great. So from the cytosol, it is going to move to this endoplasmic reticulum by using this TAP. Yes, everyone? Clear? Okay, so this is a very interesting question. So and absolutely you will get questions from this vaccines and all. Okay, so please read such questions and go. Okay, right. So what is the minimum percentage of children who needs to be vaccinated successfully to achieve the herd immunity to diphtheria? Okay, yeah, diphtheria, tetanus and all is there, no? So for that, how many percentage of them they should have received this one? Yeah, to get a herd immunity. It is 25%, 10%, 50%, 75%. Yeah, Shrishti, very good. Correct. Yes, everyone. Yeah, Shushmita. Yeah, Sunanda. Correct. Yes, Sanjay. It is going to be 75%. Why? Why the lower percentage is not going to give you the herd immunity? What about the nature of this tetanus and diphtheria? I think tetanus is the TT injection, okay? So even if 6 months is over, what will happen? What will the doctor will advise you? If at all you get pricked by some iron or glass, yes, immediately your parents will take you to the doctor and put the TT vaccine. Correct? Yes, tetanus toxoid is preventing the sepsis. Likewise, this diphtheria will also do that. Okay. So it is not, yeah, correct. So in general, the herd immunity is, the concept of herd immunity is, I'll tell you the difference between other infections and this one. Okay, yeah. So in case of herd immunity, for example, if all your neighbors are vaccinated, okay, even if you are not vaccinated, you will not get the infection. Correct? So this is the concept of herd immunity. Right? Yeah, absolutely. So but now what happens, this is in case of this diphtheria and tetanus. Yeah, so there is one more risk. So the immunity should come from individual level. It is not dependent on herd. So you have to go for the maximum one. Okay, so that is why you go for the 75% level. Okay, understood? Okay, yeah, good. So for the vaccination against this mycobacterial disease like tuberculosis, okay, the most important fact of this immune response is to be stimulated, it is going to be a higher titer of antibody. It is going to be the macrophage activated cell mediated immunity. It is the cytotoxic T cell or antibody in this gut lumen. Come on all. So which of the following is absolutely going to make it out? For vaccination against this microbacterial disease like tuberculosis, yeah. So the most important fact of the immune response to be stimulated. Yes, very good Shushmita, absolutely correct. So it is none other than your macrophage activated cell mediated community. Yes, Tanya. Yes, Ananta. It is going to be B. Okay, not C. It is B. Right? Why? In tuberculosis, why B is important? Yeah, Sati. Because it is an intracellular infection within the macrophages. Okay, within the macrophages only this TB is going to affect. Okay, so if you stimulate the T cells and they produce this macrophage activating cytokines like interferon gamma, this will be very, very appropriate. Oh yes, Nanda, come on, that is okay. It is okay to commit mistakes. Yes, then only you will be learning and learning and learning. Yes, correct. If you don't make any mistakes, your learning graph will be very low. Correct? Yeah, it is okay to make mistakes now. Correct? Yes, all. Okay, guys. So BCG. BCG. It is used to protect against which of the following? Yeah, Shushmita. Correct, no? Tuberculosis, rabies, hepatitis B, influenza. Yes. Yes, all. Come on. BCG. BCG vaccine was there, no? Yeah, Shibani? Yes, Shrishti? Correct. So it is against the, very good Meghna. Yes, Ashima, Sanjay, Anjali. Yes, Sushmita. It is going to be the tuberculosis. BCG, it is protecting against the tuberculosis. Bacilli calmette-guerin. Okay. So they were developed by our Pasteur Institute only. Yes. So did you know how did they produce this? They added the bile juice. Okay, to the culture media, and then they found that there is the dispersed growth of mycobacterial tuberculosis. Okay, but what happened? It became attenuated. So it is protecting them against. Yes, come on all. Yes, everyone. So this is all clear to you? Yeah, BCG is going to be that. Right, good. So the next one. So this is also from vaccine. Okay, all. Shall we take up this question? Yes, all. Correct. Good. A small protein subunit that is used in the vaccine may fail to stimulate the T cell immunity. Why so? Why, if it is going to be a small protein subunit, yes, it is failing to stimulate the T cell? It is because they lack this glycosylation, or they lack the conformation, yes, or they lack the carrier determinants, or the HLA related unresponsiveness is going to be there. Yes, Urvish, Anjali, Shushmita, Sati, Yashmita. Shall we check out? Very good. Absolutely. D is going to be the correct answer. Okay? Yes, correct. So HLA related unresponsiveness is going to take place. Why? Because this subunit vaccines, they are just short peptides. Okay? Yeah. So they will have many polymorphic forms, right? Yeah. So they cannot make the HLA to be properly responsible. Okay, so that is why they fail. They are not stimulating the T cell because they are not achieving this, they are achieving this MHC thing. Got it, everyone? So now this is clear to you. Okay. So when you will use a carrier, whenever your thing, it is not immunogenic, you want to make it. For example, you take the starch. Yes, absolutely. Yes, absolutely. Correct. Yes, true, Sananta. Yeah, true. Yes, Sati, it is like a hapten. Yeah, Sananta. So it is going to be HLA related only. Yes, everybody, it is clear to you? Okay, good. So shall we make a move towards the next question? Or we will revise it once? Yes, we'll just revise the MCQs one, because for many of you these points were new, right? Yes, so we'll just recap it once before we move. Okay, right, I think till this we have done. Okay, so from this question we will just revise and then we'll go. All right? Yes, everybody, come on. So this binding of antigen to this antibody, fast we will do. The binding of antigen to this antibody, which was the correct answer? It is optimized by spatial complementarity, isn't it? So that was the correct answer. Yes, absolutely, Shushmita. So if it is going to be this binding of antigen to this antibody, it is going to get optimized by the spatial complementarity. Just to check at least now if you get the correct answer or not. If not, you have to revise one more time. So that is why I am just putting the questions one more time now. Got it, everyone? So please do revise. Whenever you are studying the MCQ, certain thing you will get it correct on the first go. Okay, but in the second go also you should get it correct. Then only that means that your MCQ practice is done. Okay, if not, again you have to sit and read, right? Yeah, so which one of the following statement is incorrect about the affinity? It is the measure of strength of binding. No, I mean it is correct statement, and it states that the association constant, it is correct. Free energy change, correct. Which is incorrect? Avidity is going to be incorrect because avidity is representing the wholesome affinity. Correct. So shall we revise the next question? Okay. The processing of the cytosolic protein is involving. Right. So the processing of the cytosolic protein is involving transport into the late endosome, proteasome mediated cleavage, displacement of invariant chain, displacement of beta 2 microglobulin. Very good, everyone. It is going to be the proteasome mediated cleavage. Understood, everyone? Right? Okay, so we saw about this TAP. Right, so transport associated protein, right? And for herd immunity, it should be at least 75%, right? Because individual level is also important. And if you want to vaccinate against tuberculosis, yes, which is going to get stimulated, it is going to be none other than the macrophage activated mediate, because tuberculosis is going inside the macrophages. Okay, so BCG, it is protecting against the tuberculosis. Correct? Yeah, so that is against the tuberculosis. And the small subunit protein, it cannot stimulate, it fails. Fails means it cannot stimulate the T cell. Why? It is very small, no? So it is leading to HLA related unresponsiveness. So it is not stimulating. Clear till now? Yes? Okay, good everyone. So what about the DNA vaccines? Okay, so they are relatively poor at stimulating the cytotoxic T lymphocyte response in mice, right? Or these DNA vaccines, they must be administered on the gold particles. Okay, so if they are to be effective, all right, so they are only effective, it is followed by the protein boost. Okay, so the DNA vaccines, they have advantage when preparing the subunit vaccine against the virus, which is frequently altering their antigen. Come on everybody, a very very interesting question and important question too. So let us see how it is now. Yes, what about your DNA vaccines? Yes. Yes, all. Yeah, Shishmita. What about others? Yeah, Shrishti. Yeah, Sunanta. Correct. So it is this DNA vaccines. Yeah, absolutely. Yes, Simran. Yes, Tanya. You're all correct. Yes, Sunanta. Sati. Yes, Shrishti. Okay, everyone. Keep it up. Yes. So they have the distinct advantages. So what happens if it is going to be a protein, then you use a which vaccine? Yes, Meghna. So you use the subunit vaccine. Now the virus is changing the protein. Epitope is changed, right? So it will escape your vaccine. But if you do it as a DNA vaccine, then the DNA cannot change, isn't it? Right. So if at all the virus is actively altering the antigen, you have the only option to go with the DNA vaccines. Yes? Too good. Very good. Tetanus toxoid is usually given to humans. Okay, right? So using which as a adjuvant. Okay, right? Yes. So our which is a carrier. Okay, so it will be tetanus toxoid. It is fused with Simran. Class starts at 9 p.m. every day. Okay, yes. So till 26th. Okay, so I have this 8-day series, right? So it is at 9 p.m. every night. Yeah, Simran. Okay, good. So this tetanus toxoid is usually given to humans. Yeah. Yeah, Simran. Yeah, Shishmita. So this is going to get absorbed into aluminum hydroxide or complete friends adjuvant, or it is going to without the addition of any other agent, or it is together with the toxin. Tetanus toxoid. You require some medium, no? No, it is none other than your aluminium hydroxide, ALOH. Okay, ALOH, it is a very very important one. Okay, so what happens? This exotoxins, no? So first they will be detoxified. Okay, yeah, so this bacterial exotoxins, they will be detoxified. How? By using the formaldehyde. Okay, right. So this formaldehyde, it is not going to, yes Anushka, so this formaldehyde, it is not going to destroy the immunogenic determinants. Okay, right. What they do, they are given after absorption to this aluminum hydroxide. This is only acting as an adjuvant. Okay, so for human, we can use safely this aluminium hydroxide as a good adjuvant. Yeah, alum is an adjuvant, right? So this tetanus toxoid, it is going to get absorbed with this alum hydroxide or aluminium hydroxide. So this is the most commonly used method. Okay, yeah, adjuvant means they will be binding and they are used as a carrier. Okay, so what happens is the adjuvant, generally you have the haptens. Yeah, Yes, Arif. So you have this ICMR JRF. Should I wait for CSIR? You can go with ICMR, Arif. So there is nothing wrong with taking or claiming your ICMR JRF. It is also a very good fellowship, okay? And ICMR has many advantages too, okay? So definitely go with up your ICMR fellowship. Yes, Arif. Right. Yes. Yeah, Nirmala. So now you understand. So what happens? Certain antigens, they will not be immunogenic. Okay. So what you have to do is that you will add something and you will make them as a carrier and you get transported inside. Okay. So that is what. Yeah. Yes. Now you can join. Later you can give any exam. That is fine only. Yes. Yeah. But why to wait? Okay. Right. So because rules and regulations are changing day by day, it's better that you get yourself enrolled in the PhD as early as possible. Yes. So that is the best advice ever I will give anybody. Okay. So if at all you have a valid fellowship, join immediately, because day by day, yeah, you can try. Absolutely, if you can try for the examination, but please enroll for your PhD at the earliest. Yes, because scenarios are changing. Yeah, Sananda, scenarios are changing day by day. Okay, so it is fast the better. Okay, right. Okay, everybody. Yes. Yes, Arif. I understand. That is true. But now you got ICMR, no? So make use of it. Yes, the same thing only you will be getting. Everything else is same. Okay. So don't worry. You can, for your satisfaction, definitely you can give the exam anytime. Correct? Okay, all. Good. Yes, everyone. True. So the potential disadvantage of immunological protection using this passive transfer of horse globulin. Okay. So you are giving this horse globulin and there is going to be a disadvantage. So what is the disadvantage? What is passive transfer? You are not inducing your cells, but you are simply giving the antibody, right? Yeah. Yes, Sananda. After joining, definitely the maximum the exams you clear up, the maximum the value will be. Yes, right. But it is advisable to join the institute and then you write the exams. Yeah, nobody is going to wait, isn't it? Correct, Sananta. Good. Yes, Sushmita. It is absolutely the serum sickness. Right? No, Meka. It is going to be the serum sickness. Right? So let us see what is the serum sickness. See here, if at all you take this horse globulin, they will contain this anti-tetanus and anti-diphtheria toxins. Okay, right. Yeah, but what happens? Yes, Simran. Yeah, Sanjay. So it is going to make the serum sickness. Why? Why serum sickness develops? This is because they respond to the foreign proteins and so they will get the skin rash, swollen joints, all these immune effects will be induced. So this is what is called as the serum sickness, because it is some other organism, no, right? So that is going to induce the serum sickness. Understood? Yes, everybody. So now you got what is the serum sickness? Yes. Okay, all good. Right. So the next question, a feature of salmonella based vaccine that is expressing the antigens from other infectious agent. Okay. Yes, is that the immunity, it is limited to the gut. Only secretory IgA it is elicited. It does not invade the mucosal lining of the gut. It provokes oral and systemic immunity. So if you are using salmonella based vaccine, it is very good. Why? Why salmonella it is regarded as a good one for your vaccination? As a vector, right? So why it is so much trusted? Yeah, Anjali, absolutely correct. So salmonella will provoke both your oral and systemic immunity. Yes, absolutely correct. Yeah. Yes, Shrishti. Yes, Tanya. Very good, everyone. Yes, Anjali. It is going to be option D only. Okay. So the salmonella, it is infecting this your mononuclear phagocyte system. Okay. And it is going to produce both the secretory and systematic one. Okay, so that is why it is, yeah, so that is why it is very much preferred. Okay, so this collar and all, it is orally administered only. Correct? Yeah. Yes, Arif, absolutely. Serum sickness is due to that only. Correct. Yes. Right. So next what happens? An antibody responds to a protein vaccine. Okay, can only be obtained, right? Yes, if the molecule is linked to the carrier, if the molecule maintains yes, absolutely, yes, Ananta. So if the molecule maintains the discontinuous epitopes, the molecule is glycosylated only if the peptide bonds are maintained. Come on, guys. Yes. So an antibody response to your protein vaccine, it can only be obtained, that means this is a minimum point. Okay, so other one may maximize, but minimum the pass mark should be there, isn't it? So which is the minimum pass mark? See, think, guys. Yes, it is very simple only. What will happen if you remove this answer? What will happen? See that and give. Okay. Yeah. See, if your protein is already, it is not a subunit. They have given that it is a protein vaccine. Correct. So if the protein vaccine is already having a good weight, okay, or good immunogenic it is, do you require to link it to a carrier? It is already immunogenic. So you need not link it to a carrier at that time. Correct. So that becomes optional. Yes. Okay. So if it is containing discontinuous epitope, only then it can be inducing the response. Not at all. Only if it is glycosylated or if the peptide bonds are maintained. If there is no peptide bond, what will happen to your protein? Think. Yes, Nupur. So if the peptide bonds are not at all there, what will happen to your protein vaccine? It will become a subunit. Got the point? Yes. Now you understood the logic behind this question. If there is going to be no peptide bond between the proteins, it will degrade. Then there is only subunit, that is not essential, that is not enough to make anything. So the minimum one, it is going to be the peptide bond. Okay. So if the peptide bond is not there, it is just a mixture of amino acids. Okay. Yes, Tanya. Yeah. So if it is a simple mixture of amino acid, nothing is going to take place. Correct? Very, very logical, no? Yeah. So this is how the level of questions will be. It will be very, very simple. But you have to think on it. Got the point now? Yes, everybody. So now you got it? Okay, good, everybody. Simply you need to maintain the peptide bond. That's all. Okay, so this is a simple question, guy. Yes, come on. So the first immunoglobulin heavy chain class that will be expressed on the surface. Okay. Yes, all. Correct. Yeah, Shivani. Okay. Oh, Shivani, take care of your health. Yes, Sushmita. Yes, Sunanta, Tanya, Anushka. Yes. Okay. Right. Yes. Okay. Good, everyone. Come on. So let us complete this. So the first immunoglobulin heavy chain, yes, to be expressed on the surface of the newly produced B cell, right? It is going to be immunogenic IgA. Right? Yes, of course. Yeah, Satish. So it is going to be IgD, IgE or IgM. No doubt, isn't it? Yes, obviously it is IgM. Yesterday also we saw that, and even with this COVID detection also, what will happen? IgM will be first produced. Okay, yeah, absolutely. So that is the first one that is produced. Later only it is going to be the IgG, right? IgG, it is the second level. Got it, everyone? Right? Absolutely. So this IgM, it is the first class that will be expressed. That is going to get followed by IgG. Okay, right. Good. Yes, yes. So the very interesting question this is. The cytoplasmic region of surface IgM. Okay. Yes. Yes, Simran. So these questions are from NII only. Correct. Yeah. So the cytoplasm, these last three questions are from NII. Okay, old questions. Right. Before that we were doing with Reut. Yes. Yeah, Satish. Oh, thank you, Satish. Right. So the cytoplasmic region of the surface IgM. Alright. So this is going to consist of which of the following? Okay, so see here, just now we saw there are two regions. Okay, so one, it is going to be the membrane, other one is the cytoplasmic, then the organelles. Okay, right. So now the cytoplasmic region of this surface IgM, they will contain, okay, a single H chain constant region, or they will contain a light chain, or they will contain 110 amino acids, or they will contain only 3 amino acids. Very, very tricky question, guys. I'll warn you. No. Nobody gave the answer for this question. That is okay, guys. You can learn. Yes? No, Shishmita. It is going to be just 3 amino acids. Okay, right. Yeah, so I'll tell you, see here, if you take these cytoplasmic tails, yeah, so they will have a very, very short tails. Okay, especially, yes, especially if you take this IgM, right? Yeah, so this IgM, answer is D. Okay, so this IgM, especially, it is having a very short tail. It is only 3 amino acids. Got it? Right? So it is only just 3 amino acids. In general, it will be in the range of 3 to 28. Okay? But our fellow IgM, he is having only the 3 amino acid residues. Got it? Right? Yeah. So henceforth, answer D is going to be correct. The cytoplasmic region, yeah, the cytoplasmic region, it is going to contain a tail, and the tail is just 3 amino acid. Yes. Correct. Yes. Right. Okay, all right. So now it is clear? Yeah, it is amino acid only. Yes. Okay. Right. Oh, you read as D3. Okay. Now I got it. Right. Yes. So now it is clear? Alright. So now one numerical we will do. Before we are going into the recap session, we will do a numerical. Okay. So using only the random VDJ recombination. Okay. So from B it is going to be 40. From D it is going to be 30. And J it is going to be 6 segments, right? Yes, all very good, right? So V it is going to be 40, D it is going to be 30, J it is going to be 6, right? So what is the possible variable region? Very simple numerical. Yes, good, Sanjay. What about others? So what is going to be the answer? So what is the variable region? This is a random one. So what will be the variable region number? Yes? Okay. Alright. So it is none other than 7200. Right? So how did you get 7200? Just recombination. No, multiply. Right? 40 into 30 into 6. So you will get 7200. So that many possibilities it is there. Understood? Okay, all. So we have last 10 minutes. So what I have planned is I will just tell you the key points that you must have already revised. Okay, so if not, I'll share this PPT with you in this Telegram group. Okay, so I will yeah, you can, yes, so my Telegram group, it is Saranya Life Science at Unacademy. Okay, so you can follow that group if at all you want this presentation, right? And here I'll just tell you the key points alone. Okay, right, yes. So what is this? This is a structure of your antibody, right? Yes. So your antibody, it is going to contain what region? It will contain a heavy chain and a light chain. Okay, yes. So this is going to be the light chain. So this is going to contain an amino terminus, and this is going to be the carboxy terminus. Okay, and this region, which region? The amino terminus of the light chain. Yeah, so it is going to be the antigen binding region. Okay, it is the FAB portion, right? Yeah, likewise here also. Got it? Yes. Sure, you want to zoom this? All right, don't worry, I'll zoom this on. So now it is clear? All right. Yes, all. Okay, so this is going to be the antigen binding domain. Yes. So next, this is going to give the constant region. Okay, so that is called as the FC region, and this is the disulfide bonds, okay, that are formed, and then this is what it is called as the hinge region. Okay, right, and then here what happens? You have this CHO group, and one more thing that you have to remember is the number of amino acids. Okay, so heavy chain, in heavy chain it is going to contain, correct, so heavy chain it is going to contain 446 amino acids, right? And this light chain, it is very light, no, so light chain it is 214 amino acids. Got it? So for heavy chain it is 446, for light chain it is 214. Understood? Okay, so each heavy and light chain, it is made up of number of domains. Okay, right, good. So if you take this, it is going to contain a variable region and a constant region. So this is the domain. Okay, so this is for the heavy chain. No, this is heavy chain. Correct? The heavy chain is going to contain the variable region and the constant region. Understood, everybody? So these points you could have studied, but just revise one time, because these are all highly basic things, but it happens that after reading so many concepts, this basic you will forget. Am I right? Yes? Okay, so the domain is going to contain approximately 110 amino acids. Okay, so the domains, they will contain approximately 110 amino acids. Okay, right, yes, and then they will be linked by the cysteine. Okay, and then yeah, and this is another important experiment. What is this? Treatment with pepsin, papain, and then this one, beta mercaptoethanol. Okay, so if you treat with this pepsin, what happened? This is going to give you 2 FAB molecules. Okay, so if at all you are treating them with the pepsin, it is going to degrade here. So you will get 2 FAB molecules, and they are linked by this disulfide. Okay, here only it is cutting, right? So you will get 2 FAB molecules that are linked. Right, but if you treat them with papain, correct, yes, if you cut them with papain, yes, Ananta, I know. So here what happens? It is going to get 3 regions. Okay, so 1 FC region and 2 FAB region you will be getting, right? And this FAB region, are they linked? No, this FAB region will be independent. This alone will recap here. Yes, so if I treat my antibody with pepsin, what will happen? Yes, absolutely. So pepsin, it is going to give you 2 FAB, but they are all linked. Correct? Right. So if you treat this antibody with papain, what will happen? It will give you 1 FC portion and 2 FAB portion, and they are all separated. So they are all alone. Okay, right. If you treat them with mercaptoethanol, Yeah, what will happen? You will get everything. Mercaptan is digesting all the disulfide linkages, right? So you will get 2 heavy chains and then 2 light chains. Clear, everybody? And the fragment molecular weight is also clear to you all, right? So no doubts on this concept. Very important concept. Correct. Good. Yeah. So I think this one also we have done. Okay. Right. And yeah. So what is this hypervariable region? Within the variable region, the 3 regions, they are correct. Yes, Tanya. Yeah. No, not only immunology. So for NII, you have to read. Number 1 is immunology. Apart from that, more basics will be from biochemistry, and then they will ask more questions from molecular biology and little bit from microbiology too. Okay, right. So not only from immunology. Apart from that, you have physics, chemistry and general aptitude, and then research aptitude too. Okay, all? Yes? Okay, but you can take it. It is a very light exam, not as tough as CSIR. Okay, so this is the hypervariable region. Okay, and they are only called as the CDs, complementary determining regions, CDR. Clear, everyone? Right? Okay, good. Yes, everyone, keep it up. Right? Yes, all. And then what are these antibody mediated effector functions? So number 1, it is going to be this opsonization. Yes, so opsonization, what happens? The FC region, it is going to bind, receptor is there, no? So it is present in the macrophage and neutrophils. Yeah, Shushmita. And this complement activation, it is done by IgG and IgM. Okay, right. ADCC, what is that? Antibody dependent cell mediated cytotoxicity. So that is done only by this FC receptors that are present in the NK cells. So who will cross the epithelial layer? Very important question. This is a PYQ also. Who will cross the epithelial layer? It is IgA and IgM. Okay, IgA and M, they cross the epithelial layer. Okay, who will cross the placenta? One and only fellow IgG. Got it? Got the difference? Okay, all good. So these are all the important things. So this is the FC receptor that is present within the phagocytic cell. So this is how it eats. Absolutely. Yes, Simran, you can prepare. Okay, so here see, I read when I was giving the exams, I read this physics today and chemistry today. Okay, so this is from MTG publication. Okay, so this is your basic standard NCERT pattern things they will give. Okay, so they will give small tables and things, especially in chemistry and physics, since because you just want to revise that once, you can get that. Okay, some 40 rupees or 50 rupees one book is there. Okay, MTG publications, physics today, chemistry today. Okay, yes. Okay, so that will be very concise one too. All right. Yes, but you have to see which edition you are going to buy. All right. Yes, all. Likewise for biology, you can use this biology today. So that will give you a lot of practice actually. Yeah. Okay, good, everyone. So this is about all these things, and this is about how this yes, CC thing. Yeah, Shananda, absolutely correct. So this is how this disulfide bridges are formed here and there. And you remember IgA, it is a dimer and they are connected by the J chain. What about IgM? Do you have any idea on IgM? Yes? How will be this IgM? IgM, it is going to be a pentamer. Okay, IgA, it is a dimer. IgM, it is going to be a pentamer. Okay, yeah, they are going to be a pentamer. They are also connected by the J chain like your IgA. And IgG alone, it will cross this placenta, right? Yeah, so then only they will be released. And if you take this IgM, it is helpful in mucosomal transport, epithelial and mucosomal transport they are. Correct. Very good, everyone. Yes. And IgA, it is secretory. Okay, IgA, it is secretory. Remember that. Yes, so they are the secretory component. True. IgE, it is in this allergy. Correct. Yeah. And one more concept very frequently asked, it is about this. Yes, everybody. So this is going to be Yes, all. So this is going to be, what is this? This is going to be the idiotypic, isotypic and allotypic variation. Yes, all. Absolutely. Yes. So this, what is this isotopic? Isotopic means they have a very significant functional effect. Right. Yeah. And what about this allotopic? Allotopic means they do not have much functional effect. Okay, so there is going to be an allelic variation, right? So it doesn't have much functional effect, right? But idiotopic means there is going to be the variation in this antigen binding site. So these are the variety of variations that is going to take place. Okay, so what is this isotopic determinant? This isotopic determinants, where are they present? They are present in constant region. Yes, yes, absolutely correct. Yes, yeah, Shivani, absolutely correct. Yes, so this is going to be the constant region. Okay, right, determinants. So this is also very important. Isotopic, allotopic, it is the allelic variation. Okay, so they slightly differ between the individuals. So this has no effect on the antibody function. That is why you can donate your antibodies. Correct? Yeah, they are the allotopic only. And idiotypic determinants, where are they? They are here. This is the idiotopic, isn't it? So this mouse Ig1, it is against the antigen A. This is the mouse Ig1 against the antigen B. So difference between the, so this is the difference between these two things. Got it? Yes, everybody. I hope that this is all clear to you. All right. I think this one we have already seen regarding this B cell receptor and all. Correct? Okay, all. Oh, yes. Yeah. Okay, all. So this is all about this. Okay, fine, everyone. So tomorrow definitely we will meet back with, sure, I'll, so we will meet back with more information. Okay, right, all. So this is all about that. Okay, guys, I'll see you soon. Yes, everyone, I'll meet you tomorrow. All the very